The next question is how to predict immunotherapy response. So with the current cancer immunotherapy results is really exciting because compared to previous drugs, which work only for a very short time and the patient develop resistance, and also there are drugs that patient has to keep taking forever. Immunotherapy is kind of interesting in that it stimulates your immune system. And once the immune system is activated, it will do the job. And some patient can be cured, after say 10 shots, they are totally cured and they don't have to take the drug anymore, which is very exciting. Unfortunately, this is just only a minority. By this time, a lot of clinical trials are being conducted in different cancer types to predict immunotherapy or to evaluate immunotherapy efficacy. And so far, the ones that worked very well are melanoma, lung cancer, which remember have a lot of mutations. They look very foreign to the T cells compared to normal cells. But in many different cancer types, even though the drug has been approved for usage, only a minority of patients really responded. For example, as a single therapy for breast cancer, it only works in the ER-negative breast cancer or triple-negative breast cancer, but only works for less than 20%. In non-small cell lung cancer, it's also about 20-30% as a monotherapy. With melanoma, it's probably 30-40%. And so the big question is how to really predict response to immunotherapy, which patient will really respond to immunotherapy. Over the years, there are many, many different biomarkers that are proposed and also being evaluated in different immunotherapy trials. And so there are some kind of well-known ones. The first is T cell level, which means whether the tumor, the T cells are infiltrating the tumor. You can imagine the guard example before. If T cells are like guards, you want to make sure there are many guards already in the tumor. Maybe they are only tipsy, so you can wake it up. So if in the tumor you don't see any guards, whether or not they're drunk or not, then it's not very good. The second one is T cell diversity. As we mentioned, in order for the T cells to recognize the mutations, if the tumor has, if the T cell have different TCRs, many different TCR, just by chance, one of those TCR recognizing a tumor antigen would be a little higher. And so young patients with diverse T cells are more likely to respond better to immunotherapy. But in general, whether it's in the blood or in the tumor, people see that if you have higher T cell diversity, it's probably, at least in the blood, is a good thing. In the tumor, because it's kind of difficult, sometimes the correct T cell is already clonal expanded. And so that clonal expansion might hide some information about the overall diversity. So the result hasn't been as consistent. But overall in the blood, you want the T cells to be quite diverse. And the next one is PD-L1 levels. This one is kind of puzzling. PD-L1 is, cancer cells or sometimes macrophage or B cells also express PD-L1 on the cell surface. Is the cancer cell checkpoint to inhibit T cell from activating, to say, hey, buddy, let's be friends, no point to get alarmed. And so the PD-L1 interacts with the PD-1 to suppress T cell activation. And so most people think that if you have higher PD-L1, the patient should do worse because it means that the T cells are being suppressed. However, because we want to make sure that the cancer cells antigens have already been recognized by the T cells, and the T cells initially probably try to attack that's when the cancer cells will upregulate PD-L1. So PD-L1 is usually activated when the T cell has just started killing or secreting the interferon or secreting the granzyme and porphyrin to the cancer cell. That's when PD-L1 get activated. And so if we see higher level of PD-L1 expression on the cancer cells, this is an indication that the guards were trying at least to kill. And so you can imagine this is kind of some signs that the T cell have been activated before. And so that's kind of a very, very important biomarker. In fact, the Merck, they are the most successful in the current PD-1 antibody therapy market. And they very often use PD-L1 on the cancer cell surface level. They usually ask, does 30% of the cancer cell have PD-L1 or 50% of the cancer cell have PD-L1 protein level on the cell surface? They use that as a biomarker for predicting immunotherapy response. And then another one that is really important is also tumor mutation load. As we mentioned, if the tumor looks really foreign, it has a lot of different mutations. Some T cells will likely to see this as foreign. They are more likely to recognize this tumor and then get activated, right? So yeah, this is one study looking at immunotherapy in melanoma. And so the patient have DNA damage repair defects. So when they have MMR defects, their tumor has a lot of mutations, not just in melanoma, in say colon cancer. If the tumor has DNA damage repair defects, they will accumulate a lot of mutations. And interestingly, those tumors with the a lot of mutations, their tumor will shrink. Whereas those that have correct DNA damage repair mechanism, their tumor most likely don't respond to immunotherapy. And so you can see here, many different biomarkers are being predicted. So overall, in terms of tumor immune evasion, meaning that how does the tumor initially develop, and also immunotherapy resistance, meaning that they don't respond to, for example, the current PD-1, PD-L1 antibodies, there are a number of these different mechanisms, right? So this is a review in 2019 where they mentioned a few things. So let's first look at this first case. They are called immune hot tumors, meaning that there are T cells in the tumor. It's trying to do the killing, but then those T cells might be suppressed with exhaustion markers such as PD-L1 or TIM3 LAG3. And so then the T cells are there, they are just drunk. So immunotherapy hopefully will bring the tipsy T cells back into action. But then there are also cases when the T cells cannot really infiltrate the tumor. And so over time, people have discovered a number of factors that prevent T cell from activating. This includes myeloid derived suppressor cells, tumor-associated fibroblasts, high level of T regulatory cells, and also the M2 type of macrophage. Remember, the M2 is a wound healing. It's like, T cells don't come here. There's a wound, let it heal. Don't need to attack outside. And so, so with these suppressive cell types, then the cancer cell really cannot infiltrate. And also, a lot of these immune cells are going into the tumor through blood vessel. And so there has been, I would say, conflicting results about whether blood vessel is good for immunotherapy or bad for immunotherapy, but at least different studies have evaluated, and they do see some differences. Some say, having blood vessel is good, at least you bring the T cells into place, but other ones also say that it's bringing other, bad cells into the tumor microenvironment. There are, and then there are also factors that make the tumors cold or the T cells don't get activated, which is in this case dendritic cell, whether the dendritic cell is presenting the antigens well enough to the T cells or not. So one is the dendritic cell activity. The other is whether there are MHC presentation defects. So antigen presentation, if they are defective, for example, the cancer cell may lose MHC1, then their mutations never get presented onto the cell surface. That's preventing the T cells from recognizing the antigen. And the final case is lack of tumor antigens. If this tumor have low mutation burden, very low mutation, then they don't usually benefit from immunotherapy. And so you can see all of these different mechanisms. So far, each study only evaluates one or two. It's hard to really develop good predictor that works on all of them together. So within our lab, we also have a computational algorithm. We call it TIDE, which stands for Tumor Immune Dysfunction and Exclusion. We evaluate both T cell dysfunction and T cell exclusion. And so the algorithm uses the pretreatment tumor bulk RNA-seq biopsies to make the prediction. And based on the bulk RNA-seq, we can estimate the level of CD8 T cells that are infiltrating the tumor. We can also evaluate whether those CD8 T cells are activated by monitoring the granzyme and porphyrin levels in the tumor. And so from the tumor expression, we can tell whether this is an immune hot tumor or an immune cold tumor. So immune hot tumor are those that have enough T cell infiltrating, and you see the granzyme porphyrin there. And what we found was that if those T cells are in a fixed dysfunctional state, meaning the guard is too drunk, then immunotherapy would not be able to wake it up. But if the T cells are, is kind of immune hot, the T cells are just a little bit tipsy, maybe immunotherapy can bring it back again. Whereas for the immune cold tumors, there are a number of factors, whether the tumor have M2 macrophage, cancer-associated fibroblasts, or other myeloid suppressor cells, or maybe Tregs, or low antigen presentation, maybe the tumor is not really hot. And those will also not respond to immunotherapy because the current immunotherapy is trying to revive the T cells, but if the T cells are not even infiltrating the tumor or things preventing the T cells from infiltrating the tumor, then the T cells can't be activated and do the killing. And so we have this website and we have been accumulating a lot more immunotherapy clinical trial data, and also we are implementing additional reported immunotherapy response biomarkers. And so we have this paper that has a number of functions. The first is a biomarker evaluation feature. So if you come up with your own immunotherapy biomarker, you say, okay, these genes are important and these are bad, these are good, and you can use the weights to give it different weights or different signs. And then our algorithm will use all the published immunotherapy trials to try to see how well your biomarker is predicting the response compared to all the other ones, T cell infiltration, mutation load, PD-L1 level, CD8 infiltration, and so on. And another is a response prediction algorithm. And so if you have this is for oncologists. If you have many different patients, each with a pretreatment tumor RNA-seq, you can enter this here and use a number of these different biomarkers to try to make predictions. And so if every biomarker says that this patient is not going to respond, well, then they probably shouldn't be treated with immunotherapy. And if, so this is actually from TIDE, and you can see here what TIDE predicted to be responders indeed were responders, and the TIDE predicted non-responders were non-responders, although we do make some mistakes in here. And so if the oncologists can enter the trials, we can help with prediction as well. And so in the recent DREAM challenge of predicting immunotherapy response, we actually did pretty well in that challenge or competition. And the third one is a regulator prioritization function. This is for cancer biologists. if you do a genomic experiment, you might see many genes as upregulated, say a single cell RNA-seq, right? You see many genes that are interesting. Which one do you go? Because usually you have done expression analysis, you see hundreds of genes as being differentially expressed. Can you predict which one to be important? Because most of the single cell data are generated on a very limited system, whether it's a mouse tumor or very limited patients, we can use many other clinical trial samples or non-clinical trial, but patient tumor with survival information, and also CRISPR screens, which we'll teach in the next lecture, to help figure out which one is really important. And so to demonstrate this, recently we were in a study. We know that the glycosylation regulators are potentially important for cancer cell to evade the immune system. So if the cancer cell have a lot of sugar on the tumor surface, this can kind of suppress the T cells. And so we ask what kind of these glycosylation enzymes are the most important. And we put this into the TIDE website, and TIDE, using all the other data, help us prioritize this top gene as MAN2A1. And turns out this gene has a drug. And we were able to use all the clinical trial to see, in which trial does MAN2A1 inhibitor synergize with current immunotherapy. And it turns out it is with PD-L1. And so we were able to experimentally validate that indeed, when you combine the MAN2A1 inhibition, this small molecule drug with anti-PD-L1 antibody, it can make the tumor shrink faster. So this is kind of using all the immunotherapy data and existing biomarkers to predict the future patient response, and also potentially predict future better ways to improve immunotherapy response.